Hey guys, Jack here, and in the last video I took you through a general overview of the Th2 response, and in this video I want to talk to you about how are they induced, what chemical signals lead to a Th2 response. And again, I've got our lovely fluorescent images of horse butt tape parasite eggs. Right, here we go. So how is the Th2 response induced? Warning, this is kind of gross, so if you can't handle gross stuff, I suggest you look away. So in any Th response, a T helper cell response, we need to get an MHC2 antigen recognition. So remember, every T cell has a unique T cell receptor that can detect a unique antigen, and that antigen will be expressed on an MHC2 molecule. So we need a phagocyte, an antigen presenting cell, to eat some of the pathogen and then display those antigens on its MHC2 molecule. But you've got a bit of a problem here because nematodes and helminths are millimeters long. So a macrophage cannot eat an entire worm in order to get the antigens inside of it, in order to display the antigens, in order to activate the T helper cells. So how do you get around it? Well, this is it's so gross. This is some intestinal worms that have been treated with an anti-worm compound. So now they're being ejected out of the body. And what we can see is that these worms are surrounded by mucus, right? So the intestinal worms are constantly producing complex carbohydrates, proteins, and glycoproteins to create this jelly mess around them. Now that jelly mess is designed to protect them. It's a protective mucus that surrounds these worms, but it can be phagocytosed, and it is full of antigens that then can be displayed on the MHC2 molecule. So although the macrophages and dendritic cells, the antigen presenting cells, can't eat the worms, they can eat the mucus that comes off it and display that on the MHC2 molecules. So the first signal that we need for any T helper cell induction type is the MHC2 signal. So here we have the MHC2 molecule on an antigen presenting cell. It's presenting an antigen which came from that mucus or secreted compounds from that parasite, and it's binding to a T cell receptor here. We can see the Th3 molecules right there, and this is the T cell receptor molecule here. So this is the one with the going to do this to me anyway. So this is the one with this is the unique molecule with a unique binding domain that's specific to that T helper cell. And here we can see the CD4 molecule. Remember, the CD4 molecule recognizes that MHC2 molecule, and so that's how we know that that is a T helper cell or a CD4 positive cell that's bound to that MHC molecule. So the first thing we need to get a Th2 cell is we need that recognition right there. The next thing we need is activation of another receptor. Now, if you remember, in the Th1 response, we needed something like TLR4 activation and the releasing of an inflammatory cytokine called IL-12. And that IL-12 will induce the Th1 response. Here we need something different, obviously, because we're trying to get a Th2 response. So we need activation of different receptors. And actually, it's quite interesting. This is on the edge of our knowledge. We're not 100% sure exactly how this works. And it seems like there's lots of different ways that it works. But one way that we're pretty sure about is the binding of a mannose receptor. So a mannose receptor is a very long receptor, and mannose is a sugar. And attached to it is actually lots of binding domains that can recognize different complex carbohydrates, right? But what's interesting about the mannose receptor is it doesn't have an internal cellular domain. So activation of this receptor or binding of this receptor doesn't cause a signal to go into the cell. And you might wonder, well, how could that affect the cell's behavior if there's no signal going on? Well, it does two things. One, the binding of this seems to induce other proteins to begin a phagocytotic process. So if you bind something to the mannose receptor, you'll begin phagocytosis. But the other thing is binding to the mannose receptor seems to alter our response to activation of other receptors. So in this case, I've got TLR2, which is also activated by complex carbohydrates that come off parasites. And so TLR2 activation along with mannose receptor binding seems to induce a very specific response by the macrophage and causes the change. Now the TLR2 does have an intracellular domain, so this signal does go in through a phosphorylation cascade. And what we get in response to this, so in response to the mannose and the TLR2, is we get IL-4 release. Now what's interesting is this could actually be even TLR4, which we typically think of as inducing a Th1 response. But if you get TLR4 activation as well as mannose receptor binding, it does also seem to be able to induce this IL-4 production. So we get this production of this IL-4 cytokine, and this IL-4 cytokine is released from the antigen presenting cell. It could be a dendritic cell, could be a macrophage, and it binds to the IL-4 receptor on the Th2 cell, which then causes a bunch of phosphorylations, a whole bunch of genes to flip on and off, and it will now initiate a Th2 response instead of one of those other subtypes of Th response. So that's how we induce a Th2 response. In the next video, we're going to go over what happens once they're activated. So what does the Th2 cell actually do, and what are the downstream effects of that?